It's a pleasure to be here today to talk to you about brain health, and particularly about what I consider the biggest challenge to brain health as we age, which is the development of Alzheimer's disease pathology in the brain. So I'll divide my talk into two segments. One will be about how we're doing on the fight to end Alzheimer's, particularly how to stop Alzheimer's pathology in the brain and reduce it. And then how can we promote brain health in general with lifestyle while we wait for the miracle drugs to stop Alzheimer's to come online. First, for disclosures, these are the companies for which I consult or have a financial interest or receive royalties. Alzheimer's disease, as you know, is the most common form of dementia in the elderly, with over 7 million patients in the US. In fact, 1 in 9 people over 65 have this disease. But what's going to be most important for this lecture is to know that Alzheimer's brain pathology begins decades before cognitive symptoms. And as I mentioned earlier, Alzheimer's pathology in particular is the most common form of age-related brain pathology. If we all lived long enough, let's say to 120 years old, we'd probably have enough accumulation of Alzheimer's pathology, with a few genetic exceptions, to get this disease. If you ask how many people in this country alone are asymptomatic for Alzheimer's. In other words, they have the pathology of Alzheimer's in their brain, but don't yet have clinical symptoms. Sometimes this is called biological Alzheimer's, or pre-symptomatic Alzheimer's, or preclinical Alzheimer's disease. It's probably about 20 to 40 million people in this country alone that would be in this category. It's a very expensive disease. Patients cost the US $500 billion per year. Of course, age is the number one risk factor, then family history, stroke, brain injury, and sex. Women make up 2/3 of Alzheimer's disease patients. And given that the fastest growing segment of our population is 85 plus, it's very scary to know that 30 to 40% of those over 85 have clinical symptoms of AD, never mind how many have the pathology in their brains preclinically. And this number of cases will triple by 2050, so the cost will basically essentially crush healthcare if we don't do something about it. So I like to call this the elephant in the room. The fact is, we don't clinically diagnose Alzheimer's disease and other neurodegenerative disorders until the brain has already deteriorated to the point of dysfunction. We don't do that for other age-related diseases like diabetes and heart disease. But we need to do the same for Alzheimer's as we do for heart disease. If we diagnosed heart disease the way we diagnose Alzheimer's, we probably wouldn't do so until you had congestive heart failure, where the organs are already under stress and not performing correctly. So we need to bring the brain to where the peripheral organs are in terms of treating age-related disease. Now, the leading hypothesis for the cause of Alzheimer's disease is the amyloid hypothesis. This goes back to 1906, when Dr. Alzheimer described in his patient August Dieter plaques and tangles and neuroinflammation. But the big break didn't come till 1984, when George Glenner found that the amyloid plaques are made up of a protein that he named the amyloid beta protein. And the next break came in 1987, when as a grad student at Harvard Medical School and others discovered the gene that makes the amyloid beta protein, which I named the amyloid beta protein precursor protein, or APP. And this turned out to be the first early onset Alzheimer's gene with mutations. And it was followed by the presenilin genes, presenilin 1 and 2, which we found together with St. George Hislop and Schellenberg, presenilin 1 and presenilin 2. And these make up the 3 early onset FAD, or familial Alzheimer genes. They guarantee the disease by 60 years old. These are fully penetrant autosomal dominant mutations. They only account for maybe 1 or 2% of all of Alzheimer's. So the amyloid hypothesis in a nutshell has amyloid leading to tangles and inflammation, cell death due to tangles causing more inflammation, and then you get this vicious cycle of a feedback loop shown here. But it's important to know, as I mentioned earlier, that Alzheimer's pathology begins 10 to 30 years before clinical symptoms. These are just some of the key figures from key papers showing how here on the left, the amyloid plaque deposition is beginning 15 to 20 years before. The biomarkers for amyloid in the brain begin 30 years before. And tangles basically are commensurate with amyloid, although it might take a longer to see those by imaging. And then inflammation is the big killer that leads to neurodegeneration. So I like to put it simply like this. This is a slide I use for lay talks, that amyloid's like a match that lights the brush fires of tangles. Brush fires because they spread, they propagate in a prion-like manner. And then at some point, this causes enough neuroinflammation, which I call the wildfire or the forest fire. And importantly, whereas you might have 1x cell death here with plaques and tangles, this then triggers neuroinflammation, which is causing 10x cell death. And then this is really where you go down the slippery slope to cognitive impairment and dementia. So this is a quote from Randy Bateman, my colleague in St. Louis, who said, and I agree with this, the faster beta amyloid accumulates in your brain, the sooner you get AD. Now, there's been a lot of controversy about amyloid because in the beginning, when we put the APP and presenil and early onset gene mutations in mice, you got A beta deposition, but you didn't get tangles. Well, now we know that mice can't make tangles. They don't have the right forms of tau. So it was much ado about nothing. In our 3D human brain organoid models of Alzheimer's disease that we published going back 10 years now, we could show in human neurons that A beta oligomers lead directly to tangle formation and synaptic degradation, something we couldn't see in mice. So if you follow this ribbon diagram here, we have an amyloid forming oligomers inducing tangles and cell death. Microglial cells are trying to keep up and clear amyloid. There's also enzymatic degradation of A beta. But if these microglial cells trying to clear amyloid are overwhelmed, or if there's too much cell death and there are too many signals to the microglial cells that neurons are dying, these are called DAMPs. The microglial cells basically, quote unquote, assume there's an infection. They're evolutionarily programmed that if they uptake a microbe or even uptake a dead neuron or debris of a dead neuron, they are now triggered to now, instead of being housekeeping phagocytosis-like molecules, they start secreting pro-inflammatory cytokines and chemokines. This causes reactive astrogliosis. Now you have inflammation, oxidative damage, cell death, and at this point, you see the clinical symptoms. So the nutshell here is treating amyloid and tangles is something you do to prevent this disease early. By the time we diagnose this disease, there's already been enough inflammation down here to cause enough cell death to get to symptoms, and this is a different treatment plan here. So I also just want to mention that based on the work of Teresa Gomez-Ayala at Mass General, that if you only have plaques and tangles, basically you don't get dementia. If you look, this comes from resilient brains, where you see very rare cases of folks who die with lots of plaques and tangles. But if they don't have neuroinflammation and pro-inflammatory cytokines, they don't get dementia. So going back to this diagram here, it's really the neuroinflammation that causes enough cell death to get this disease. The plaques and tangles initiate that process. And I might say that tangles can be induced by headbangs, CTE, playing football. And even there, it takes decades before you might see dementia, which is again driven by the massive cell death and synapse loss caused by neuroinflammation. So that's the basis. Now, what do we do then? How do we treat Alzheimer's? Well, our best bet's going to be early prediction based on family history and genetics, early detection based on biomarkers and imaging, early intervention. We now know that there are over 100 Alzheimer genes based on GWAS. These are the first genes I talked about, the early onset genes. ApoE, as you know, is the late onset risk factor, the most common late onset risk factor. One copy of ApoE epsilon 4 variant increases risk by 4-fold. 2 copies increases risk by 14-fold, but they're still not fully penetrant, at least not in our normal lifespan at this point, as are the early onset genes, where onset is by 60 years old or often by 50 years old. If we all live to 120 years old, probably E4 variant in 1 or 2 doses would look like it's fully penetrant. And then you have a whole slew of genes that come out of genome-wide association studies, or GWAS, where these have relatively low effect sizes in terms of effects on risk, but they're much more common. So you can see that the early onset genes are rare, but are fully penetrant. ApoE is relatively common with very high penetrance, especially with 2 copies. And many of these GWAS hits, which I'll get into, are common, but have low effects on risk. Okay, so how do we get then? And I should mention that right now, despite some claims, we don't have a real reliable polygenic risk score for all these genes together. Nothing really beats ApoE alone. If you compare all the genes from GWAS together as a polygenic risk score versus ApoE alone, ApoE is still these are not, it's still not significantly better than just an ApoE4 risk assessment. For early detection, we have imaging for amyloid and tangles available, also imaging by FDG PET for neurodegeneration. We have now blood tests for amyloid based on looking at the ratio of A beta 42 to 40. This is the amyloid beta protein, the longer form 42, which is more amyloidogenic versus 40. And in the blood, we can look at a tangle protein called p-tau217, which is specific to amyloid-induced tangles or amyloid-induced tauopathy. And I might add that the reason what you see in the blood is if the A beta 42 to 40 ratio is low, it's indicative of amyloid in the brain because A beta 42 is very sticky. And if there's lots of amyloid in the brain, the 42 can't get out, whereas the 40 can still exit pretty well. So the ratio of 42 to 40 goes down in the blood, indicative of amyloid in the brain. And that amyloid is inducing tangles where the main epitope is this p-tau217. It's a phosphoepitope. And so this is a test available, for example, by C2N in St. Louis. Neurofilament light chain is a blood-based biomarker for neurodegeneration. And in CSF, you can look at these same things as in blood, a little more sensitive. But of course, the hope is to be able to do in blood as well as we do in cerebrospinal fluid. Now, most importantly, if you find out you're in trouble, what do you do about it? What do we have for early intervention? And I want to focus up here on the prevention stage here, where you have A beta production leading to amyloid. So one idea is, can you block A beta production? And here, I would call this secondary prevention. You already have amyloid formed in the brain, but can you get the amyloid removed from the brain as secondary prevention? So let me talk about that. So I like to talk about amyloid accumulation in the brain like a kitchen sink overflowing with water, the brain overflowing with amyloid deposits, okay? So there are 2 things you can do. You can turn off the tap or turn off A beta production, or call Roto-Rooter, unclog the drain, clear the A beta from the brain. Now, in terms of trying to block the A beta production, there's been failure. Unfortunately, pharma companies went ahead and tried to completely inhibit the 2 enzymes that clip APP, beta secretase clips here, and then the gamma secretase clips here. These are the 2 enzymes that excise amyloid beta peptide from the precursor protein shown here. Well, it turns out the gamma secretase and beta secretase have many other substrates. And so if you just hit them with a sledgehammer and inhibit them, these patients don't do well. Those trials failed due to safety in both cases. So about 20-something years ago, my late colleague Steve Wagner and I took advantage of the information that most of the mutations that cause early onset familial AD, APP and presenilins, don't increase amyloid beta production, as you see in Down syndrome or with what's called the Swedish mutation. Over 500 mutations in these 3 early onset genes increase the ratio of 42 to 40 in the brain. So the 42 to 40 ratio goes up in the brain. This drives more amyloid. And going back to that biomarker in the blood, when there's more amyloid, the 42 can't get out of the brain. It's too sticky. So now it's the 42 to 40 ratio going down in the blood that's indicative of amyloid in the brain. It's a little counterintuitive, but these mutations increase 42 to 40 in the brain. So what we developed going back 20 years ago is a gamma secretase modulator, not an inhibitor. This is an allosteric modulator drug that basically allosterically opens the mouth of gamma secretase to allow the APP fragment that's already been cleaved by beta secretase to fit in the mouth better. And when you do that, you get less of the more amyloidogenic toxic A beta 42 and more of the smaller A beta 38 that's actually been shown to be protective. Importantly, you don't inhibit gamma secretase at all. So gamma secretase can hit all its other substrates like Notch and other membrane proteins that need to be removed by gamma secretase. So you eliminate that side effect that was seen with the gamma secretase inhibitors. This is the last 25 years of development of our gamma secretase modulators, or GSMs. It goes back to a company I started in 2001 that went bust in 2010. Then since 2010 at Mass General and at UCSD, collaboration there, we've been developing thousands of analogs of these molecules. Cut to the chase today, thanks to the Neurotherapeutics blueprint from the NIH and funding from foundations like the Cure Alzheimer's Fund. We have a lead candidate ready to go into phase 1, ready for an IND. For this purpose, I founded a new company called Active Pharmaceuticals to then sponsor that IND. And now we're in the process of submitting that IND. Hopefully we'll be in phase 1 then this year. So that's the gamma secretase modulator for turning off the tap. How about clearing A beta from the brain? Well, here there's good news as well, although there's a bit of a concern, and that is that we have Leqembi and Kisunla, the two new approved Alzheimer drugs. These are immunotherapies. These are antibodies against amyloid beta, Leqembi from Eisai and Kisunla from Lilly, approved in 2023 and 2024. These amyloid immunotherapies get into the brain, they bind A beta, and then they activate microglial cells around the amyloid deposits to clear the amyloid. And if you treat the earliest stage patients and do a big enough trial, you can see that by removing amyloid in the earliest stage of cognitive impairment in these patients, you get about a 25 to 30% slowing of cognitive decline. So this is enough proof of concept to say it's a good idea to remove amyloid, but optimally, we'd rather not do this as early stage treatment. We'd rather do this for prevention. In fact, up to 40 million Americans may be amyloid positive right now, that biological Alzheimer's, and would benefit from removing the amyloid. The problem is these two drugs are too expensive. At over $50K per year, you have to have MRIs. That adds to the expense to make sure there's no vasogenic edema or brain swelling or microhemorrhages, which occur in 25 to 40% of patients. And in fact, in the ApoE4 individuals, it's even more profound. In fact, at Mass General, E4 homozygotes at Mass General and Brigham are excluded from this treatment because they're so susceptible to these side effects. But what if we had drugs that could clear amyloid for secondary prevention in preclinical patients who only have the pathology but don't have symptoms yet, and you want to clear the slate, clean the slate, and get rid of the amyloid? Are there safer and more affordable alternatives? How can we do that? And the key is to look at microglial cells. And microglial cells have 3 roles, and they can be a nurturer and a housekeeper. They clear amyloid and debris. But if they start eating microbes, they think there's an infection. That's called a PAMP. But if they eat dead cells or debris from dead neurons or dead cells, that's called a DAMP. They still think there's an infection. And they may not, even without the infection, they're now activated to, instead of being housekeepers, they now start inducing neuroinflammation by releasing a storm of pro-inflammatory cytokines and chemokines. So the question is, how can we do this? How can we get these microglial cells that are all upset, inducing inflammation because they ate some neurons that died as you get older due to plaques and tangles? How do we get them back to going, keep calm, carry on, and being housekeepers and clearing amyloid again? In other words, can we reprogram microglial cells? And I just told you that the new Alzheimer drugs do this. They help them clear amyloid and debris. We'd like to see drugs that can do both, clear the amyloid, but also have them be less neuroinflammatory, which causes the bulk of cell death leading to dementia. So Nature just recently published a dozen or so of the key milestones in Alzheimer's disease, going all the way back to Alzheimer's 1906 lecture. And it was interesting. This is just shows some of the things they've had. Amyloid, I showed this, the A beta being the main component of amyloid, and APP, the first Alzheimer's gene, and the presenilin gene shown here. But in 2008, they noted that we had published a paper showing the first microglial Alzheimer's gene, CD33, and that was followed by TREM2. And this really opened the door toward microglial genetics being so important for the neuroinflammatory aspect of Alzheimer's, but also for the aspect of how does amyloid get cleared in the brain, microglial cells, the housekeepers. And here's CD33, the first of these microglial AD genes that we published in '08 and was confirmed in 2011. And when CD33 is turned on in microglial cells, it basically stops them from clearing amyloid and being a housekeeper and turns them into these pro-inflammatory killers that induce inflammation. And since CD33, there's a whole slew, the fastest growing category of Alzheimer's genes are associated with microglial cells and their role in innate immunity and neuroinflammation as a host defense in the brain. But if that host defense is in excess, you have neuroinflammation, which causes excess cell death, leading to dementia. So how do we do this? Well, in our lab, as I mentioned earlier, we developed Alzheimer's in a dish, where we can basically take what takes 10 to 30 years in a patient, going from amyloid and tangles and inflammation to finally enough cell death and synapse loss to get the clinical symptoms of Alzheimer's. That can take a decade or 2 or 3. We get that to happen in a dish or a 96 well plate in 6 weeks. And this was the brainstorm of Do-Yeon Kim and others in our group, where we can have the plaques and tangles made here by neurons, the astrocytes are here, the microglial cells come in, and you can get neuroinflammation. Well, we can use this system to do drug screening now 10 to 100 times faster than in the old days, where we had to test one drug at a time in a mouse, and each experiment took a year. Now you can test hundreds of drugs at a time in 96 well plates in 6 weeks and say which ones stop the plaques, which ones prevent the plaques from inducing tangles, which ones stop neuroinflammation or get the microglial cells to clear more amyloid. So that's what we did, and we focused on reprogramming microglia. How can we get microglial cells with combinations of known drugs and natural products being screened hundreds at a time, where we can get these to take what's already on the shelf and get these microglial cells to clear more amyloid beta by phagocytosis and release less pro-inflammatory cytokine and chemokines that lead to inflammation. So I call this reprogramming microglia. And like I said, we basically screened every known approved FDA drug and thousands of natural products over the last 5 to 7 years to see which ones work best. This is the work of Do-Yeon Kim, Anna Grichuk, Luisa Kinti, and others in our lab. As a result, after all this time and spending a few $1 million of funding, we identified over 300 FDA-approved drugs and natural products that reprogram microglia to clear more amyloid and or induce less neuroinflammation. And the nice thing here is if you're thinking about clinical trials, repurposed drugs that are already known to be safe can get to patients in 1/3 to half the time of new experimental drugs from pharma that have to make it through a lot of safety testing. We are also focusing on combinations. In other words, we want to take known drugs and natural products and combinations that do both things. So we can choose from 136 different drugs and natural products that we found that induce the microglial clearance of A beta, shown, which is here. We have 105 different candidates among known drugs and natural products that prevent the microglial cells from releasing pro-inflammatory cytokines and chemokines when they're activated by the presence of dead neurons and degrading synapses. So we want to take some from this bucket that say eat more amyloid and combine those with the ones from this bucket to say less neuroinflammation, stay chilled out, don't release your pro-inflammatory cytokines, and try to come up with a repurposed combination of drugs and natural products that can do what Leqembi and Kisunla do, the new approved drugs for Alzheimer's, but do it better, not just clear amyloid, but keep the microglial cells from inducing neuroinflammation, which causes the bulk of cell death and damage. As an aside, we also found 81 drugs and natural products to prevent the A beta oligomers. Before you get the plaque, the A beta is oligomerizing these the oligomers that induce the tangles, as we've shown in our dish model. And we have 81 different drugs and natural products that prevent the oligomers from inducing the hyperphosphorylation of tau and tangle formation, which I won't talk about here. So taking these different hits now from the Alzheimer's in a dish, what we plan to do at the McCance Center for Brain Health is clinical trials that will be quick go, no-go proof of concept trials. In other words, looking for evidence that we can do in a patient what we saw in the dish. So if we focus here on these drugs and natural products that get the microglial cells to eat amyloid, similar to what the Leqembi and Kisunla, the two Alzheimer drugs that were approved, do, and combine those with drugs and natural products that prevent the neuroinflammation by inhibiting microglial release of pro-inflammatory cytokines and chemokines. You can put these in patients in small trials and then use blood-based biomarkers or CSF, or if you have the budget, brain imaging as well, and say, we're not looking for effects on cognition yet. We're just looking at the pathology. Can we see that there's pharmacodynamic evidence for engagement, target engagement, that these same drugs that got microglial cells to clear amyloid or stop pro-inflammatory cytokine release and neuroinflammation, based on blood-based biomarkers for A-beta amyloid and tangles, neuroinflammation, et cetera, can we see that they're working in patients? So these trials end up being about maybe somewhere around 40 patients. They're trials where the patients act as their own controls. In other words, you look at a lead-in period for the biomarkers, and then you switch them to drug and see what the biomarkers look like. Each trial costs about $2 to $2.5 million, and we want to take as many shots on goal as we can. We plan on testing at least a couple of dozen combinations in these categories to see which ones might work. And this just to finish up this part of the presentation, I told you about the gamma secretase modulator for A-beta production. These are the two approved drugs for inducing clearance of amyloid, Leqembi, Kisunla. There's also evidence that 40 hertz stimulation can get this to happen. I have RD, these are repurposed drugs that we have that do this. We have repurposed drugs to stop tangles subsequent to amyloid. We also have data that a muscle hormone or a myokine called irisin, discovered by Bruce Spiegelman, we showed can induce astrocytes to enzymatically degrade amyloid by releasing an enzyme called neprilysin. And down here, where we have the microglial cells releasing cytokines, like I said, we have repurposed drugs to stop this. There are also folks who have looked at things like TNF-alpha receptor antagonists and immunotherapy to this process. But a lot more going on up here than down here right now. So the hope for early prevention is probably much higher right now than our hope for stopping neuroinflammation and cell death, although this is how you might really want to treat a patient who's already clinically diagnosed. Now, for the last part of my talk, I want to ask, what can we do now to reduce risk for Alzheimer's disease while we wait for these drugs to come online? And I developed an acronym I call SHIELD, based on what you can do to best protect your brain, meaning how can I, with my lifestyle, encourage the removal of amyloid and reduce neuroinflammation. Sleep is the S. It's during sleep that you actually clear amyloid and other debris from your brain. During sleep, the glymphatic system is turned on. Every time you have a sleep cycle, I call it a rinse cycle for the brain, you're getting your glial cells to clear amyloid. Even a good nap where you might have some REM and a little bit of deep sleep, drool a little bit on your desk, is enough to remove some amyloid from the brain. So sleep 7 to 8 hours a day, take naps if necessary. H is handle stress. As you know, stress leads to cortisol production. Stress is an inducer of neuroinflammation. So we talk about handling stress, things like a meditation practice, or just learning to relax a few times a day and focus on your breathing. Anything you can do to just counter your expectations of yourself and others in social media and emails or in my case, in my lab, expectations of my students, just kind of chilling out. Handling stress is important. Interaction with others. So loneliness increases risk for Alzheimer's by twofold. That means you're alone and you're stressed out about it. Some people like to be alone. but interaction with others can also be very helpful. Now, exercise has been increasingly important, and you want to aim at about 150 minutes of moderate exercise. It doesn't have to be great. Just a brisk walk or a ride on a bike. When you increase blood flow, and I mentioned this earlier, when you increase blood flow to the brain with an increased heart rate, this delivers that muscle-derived myokine, irisin, to the brain. It then binds to astrocytes and induces an enzyme called neprilysin that is known to dissolve amyloid. And which the nice thing is since you can actually give irisin peripherally, and it gets into the brain. So there are trials being planned on that. We also showed in a paper in Science a few years ago and confirmed it since that exercise triggers the birth of new neurons in the hippocampus. This is called neurogenesis. When you exercise, you not only get neurogenesis, which is not enough, but you also induce BDNF, brain-derived neurotrophic factor. And it's that combination of BDNF that helps keep these newborn neurons in the hippocampus through neurogenesis alive. So we showed you have to have both neurogenesis and induction of BDNF to mimic the effects of exercise. And the really cool thing is in the Alzheimer's mice, in that paper from Se-Hoon Choi and our group in 2018, we showed that when you induce neurogenesis, even if there's already amyloid there's pro-inflammatory cytokines, there's inflammation, the whole battleground is still there. But in the presence of neurogenesis and BDNF, the mice improve cognitively. And this is important for patients who already have the disease, where there's already abundant plaques and tangles and neuroinflammation. Because if we can induce neurogenesis, keep those new neurons alive with BDNF, with pharmacological treatments, well, that could be a way to perhaps, if we see in them what we saw in the mice, affect cognitive improvement, even in the presence of excessive amyloid and tangle and inflammation pathology. So that's exercise, the E. L is learn new things. Okay, so the bottom line is Alzheimer's, the degree of dementia in Alzheimer's over the age most closely correlates with loss of synapses. So we talked about how you lose neurons and how you lose synapses with plaques and tangles and neuroinflammation. But the bottom line is that the more synapses you make, it's like money in the bank. The more synaptic reserve you have, the more synapses you make, the more synapses you can lose before you get into trouble. So learning new things. Right now, if you're learning something new with my speaking, then you're protecting yourself against Alzheimer's. If I'm putting you to sleep right now, I'm still protecting you from Alzheimer's disease. Either way, you win. So that's learn new things, the L. Now, diet is, I think, most important because it's never been more true. You know, you've heard the saying, what's good for the heart is good for the brain. I think it's equally true to say today that what's good for the gut is good for the brain. We have seen that diets that are good for the gut microbiome, those 5,000 or 6,000, maybe some say 8,000 strains of bacteria, trillions of bacteria that live in your gut, producing metabolites, short-chain fatty acids that are good for the brain. Your gut microbiome is one of your best allies for keeping the brain healthy. The gut microbiome likes plant-based foods, fruits, veggies, nuts, vegetables, leafy greens, whole grains. These are high in the Mediterranean diet. We call these things prebiotics. Anything that's crunchy, that's not a potato chip or a candy or a French fry, that's plant-based. It makes it by balancing the gut microbiome. And there's a certain ratio in the gut microbiome of Bacteroides to firmicutes. And this gets reversed in the wrong direction in Alzheimer's disease. And it actually prevents the clearance of amyloid in the brain. And so I don't have to, it's a whole lecture in itself, but balancing the gut microbiome, starting with diet, is a great thing to do. So we have published several papers with Sam Sisodia and Dave Holzman and others showing that if you alter the gut microbiome of Alzheimer's mice, you can induce microglial clearance of amyloid quite readily and reduce neuroinflammation through the gut. So we kind of we put SHIELD into action recently in a small trial we did with Dean Ornish, and this is also with Stephen Arnold at Mass General. It's small. It was a phase 2 trial, Alzheimer patients, about 50 patients. that were confirmed with Alzheimer's based on their blood plasma-based A beta 42 to 40 ratios. And it was an intensive lifestyle intervention to see what we could do based on SHIELD and based on also the work of Dean Ornish. And it included probably most prominently a whole foods, minimally processed plant-based or vegan diet. Dean actually had these meals made at a facility and delivered to the patients. 21 meals per week plus snacks, enough for the patient and their spouse. This is a diet that was meant to really aid the gut microbiome. We supervised sleep. We encouraged interaction with group sessions of Zoom sessions, interacting, talking about whether they hated or loved their plant-based diet. There was supervised exercise, lectures on brain, lifestyle. I invented this device some years ago, about 10 years ago, light flashing with sound that brings you down to a theta frequency, 7 hertz, which is a relaxation frequency. It's a way to induce a meditative-like status in a patient by slowly entraining their brain in over 10 minutes from the alpha beta, where they are now with attention, down to 7 hertz theta frequency for relaxation. So they all got these glasses, which they loved, to get a kind of an instant meditation type of experience. And here's what happened after 20 weeks. CDR sum of the boxes, the most challenging cognitive test to hit. We slowed cognitive decline as much, if not more, than the 2 newly approved Alzheimer drugs. On the CDR global component of CDR sum of the boxes, there, some people actually got better. Lower is improved. They actually didn't just go down less, they didn't decline more slowly. Some of them actually improved. And you can see the placebo group here, again, this line going up means they're getting worse. And then here's the CDR sum of the boxes where it's flattening out a little bit. So you're greatly slowing their cognitive decline. Here they're getting better. Now look, it's a small trial, but it says we should keep going. In fact, we also saw that the blood A beta 42 to 40 ratio, it was indicative of amyloid removal from the brain with a significant p-value. And Rob Knight did the gut microbiome work and showed a very positive change in the pattern of the gut microbiome, where beta hydroxybutyrate, a short-chain fatty acid, was particularly increased. So at the McCance Center for Brain Health, Jonathan Rosant developed the Brain Care Score, which takes into account, it's a questionnaire that takes into account SHIELD, but also asks about your blood pressure, your blood sugar, cholesterol, BMI, et cetera. And you get a score from up to 21. And now the Brain Care Score has been introduced into EPIC. Jonathan's rolling this out beyond the McCance Center now globally to look at all these brain care score areas. So this is the first way in which people can start taking into account, am I taking care of my brain? How much alcohol? What's my nutrition like? Smoking, aerobic activity, sleep, stress, social relationships, keeping track of my numbers. So the brain care score is trying to encapsulate that. So I'm going to end now with this recommendation for lifestyle, very simply. Check your numbers, check your lipids and your A1C and your blood pressure, your BMI, get sleep, relax more, socialize more, move more, learn more, eat healthier. And, because genes matter, choose your parents wisely as well. Genetics does matter. So I'm going to stop there. I'm going to acknowledge the Genetics and Aging Research Unit, where all the lab work is done that I talked about. These are our professional staff and many talented technicians and students. Also the McCance Center for Brain Health, where we'll be carrying out these trials of these combinations of known approved drugs and natural products to come up with a safer, more affordable way to carry out secondary and even maybe primary prevention of Alzheimer's disease. My funding comes from the Cure Alzheimer's Fund, the NIH, JPB Foundation, and Rotary. And I'll stop there and thank you very much for your attention.